Welcome to today's webinar with the title 3D CoSeedis Chips, a new standard in 3D cell culture. Before I'm going to present the advantages that 3D CoSeedis can actually offer in 3D cell culture, I would like to address the simple but nevertheless important question, why we are actually interested in 3D cultures to start with. Classical 2D cultures are done in monolayers, as you know. However, the past decades have shown that cells growing in 2D on a hard artificial surface normally show structural rearrangements of their cytoskeleton. These rearrangements then affect the composition of cell surface receptors, which again has a severe influence and alters metabolic pathways. It changes the response, therefore, also to applied compounds, and as a consequence, causes false positives and negatives in according test setups. So the question has always been, okay, now that we know all those disadvantages, is there a way that we can overcome those and provide a system which is probably physiologically more relevant? And of course, the simplest answer to that question is yes, let's try and grow cells in 3 dimensions. So let's apply 3-dimensional cell culture technologies. In this context now, cells grow in an environment that resembles more their natural physiological surroundings. They do not artificially remodel their cell structure. Each cell is in fact surrounded on all 3 dimensions, so to speak, by neighboring cells. It can communicate with those cells, and because it doesn't remodel the cell structure, it shows a more accurate response to test compounds. On the graph here shown, you can see the example of a microtumor, which either has a centralized blood vessel or blood supply, or a microtumor where the blood vessels are localized in the periphery. And as you can see, it's very easy to acknowledge that the cells facing the blood vessels get, for example, the highest oxygen concentration, whereas the further away a cell is from the blood vessel, the lower the oxygen content. And that results in a microtumor quite often in a necrotic core. The same we can mimic now in vitro with a microtumor or spheroid, where we observe similar effects and build up similar gradients. However, so far there have been severe problems to 3D cultures, and those problems have included that some of these technologies to build these spheroids are relatively complicated, and they're certainly very hard to upscale to make 3D or to produce 3D spheroids in high enough numbers. Similarly, a lot of these systems are unreliable and very hard to reproduce, and that's simply because they have not been standardized to a level that would allow reliable and reproducible production of 3D spheroids. So there are massive variations in the uniformity and homogeneity of those spheroids on microtumors, and that makes it consequently very difficult to implement those tumors in applications such as high-throughput screening, high-content screening, but also some clinical applications where it's, for obvious reasons, very important that the tumors you look at, or the microtumors you look at, are very reliable, very uniform, but also very homogeneous in their composition. And that's exactly where 3D CoSeedis now comes into play and offers benefits to all existing technologies. So 3D CoSeedis is a very nice tool to mass produce highly homogeneous and uniform organoids or spheroids. It is therefore capable and shows high reproducibility. It has a unique distance co-culture design, which I will address on the next few slides, which consequently allows us also to address a wide range of disease models. And because it is an easy mass production tool and of those highly homogeneous and uniform spheroids, it is ideally suited for HTS and HCS applications. Now, what does 3D CoSeedis actually look like? And first of all, I would like to introduce you to our 3 chips. We have currently 3 different formats available of the 3D CoSeedis. We named them chip 880, chip 200, and chip 680. The number represents always the number of spheroids, microtumors, or organoids that can be grown per chip. And as you can see, 2 of those chips have been designed for a 6-well platform. The chip 680 is designed for a 24-well platform. It has been therefore our goal from the very beginning to build a very flexible and versatile setup that is not only adjustable to throughput, but that should also give the researcher a lot of possibilities to adapt to different cell sources and cell types. The system is ideal for multicellular biological 3D models, so we can form, in fact, organoids. But we can also mimic organ communication, something I will address later on when I'm gonna talk about the use of feeder cells in distance or in contact, for example. This distance co-culture conditions are also the reason that we can later on work under defined conditions. So it is possible, for example, to grow cells in the absence of any serum in 3D CoSeedis. And also from the very beginning, we were aiming to create a system which would allow us long-term cell cultures. Now here, for example, is some data shown from a publication where the researchers have looked at the combination of hypofractionated irradiation and hypothermia on microtumors. And there are 2 things you can see. First of all, in order to pick up any differences between those 2 treatments, it is necessary to have the microtumors in cultures for up to 70 days. At the same time, you also see that the differences between those 2 treatments are relatively minor, which indicates that they are stochastic, that we're talking about stochastic and rare events here. So it is necessary to expose hundreds of microtumors to the very same conditions simultaneously in order to pick up those differences, which of course make a huge difference as well for a patient suffering from such tumor. So we have 2 aspects here. One is long-term culture, and the second one is that we can expose many, up to 880 tumors to identical conditions and therefore get very good statistical relevance. And of course, this multitude of microtumors that can be produced now is also an ideal tool for high throughput and high content applications, about which I'm going to talk more in the next slides. Now, the unsurpassed uniformity and homogeneity was, as I indicated, a goal from the very beginning when we designed and developed 3D CoSeedis. We have achieved now a very high grade of reproducibility and comparability, not only between individual microtumors in one chip, but also between different chips, or even chips grown in different 6 or 24-well platforms. We have a very reliable system setup. Handling of our chips is really easy, and we have a compatibility with high throughput and high content screening. One aspect of the compatibility to high content screening is also the fact that our system supports automated image analysis. Again, in our application sections, I will talk more about this. Now, how does the 3D CoSeedis chips look like? Up here on this slide, you can see some electron micrographs on the chip surfaces. These were taken from the chip 880 or the chip 200. And as you can see, the major difference between those chips is basically the distance between two microcavities within the chip itself. The chips are made of agarose, so they are therefore don't allow cell adhesion, which is the reason why the cells, when seeded on the chips, will tumble and stumble down here into the microcavity, where they are forced to, by gravity, to form an aggregate and eventually a 3D spheroid, for example. And the fact that it's made of agarose is also responsible that we can use those chips to maintain stem cells or iPS, for example. And experiments have even shown that it's possible to induce later differentiation of those cells within the chip itself. Now, our particular distinction to other technologies, however, and that's also where our proprietary knowledge lies, is the unique geometry and topography of those microcavities. They are responsible, for example, also that now cells do grow into 3D aggregates that would normally not do so in alternative technologies. We've picked here one cell type, which is called miopoca. It's a pancreatic cancer cell line, which does not form any aggregates in hanging drop and only very loose and irregular cell aggregates in liquid overlay. However, in 3D CoSeedis, they form what we call a so-called conical aggregate. These conical aggregates, they do stick together, so the cells will attach to each other, indicating that they do express to some extent extracellular matrix components. They show all the characteristics that we see in a standard spheroid microtumor, namely that we have a proliferative outer cell layer surrounding a necrotic core if the conical aggregate gets large enough. And last but not least, we can also harvest those cones so they remain stable even when harvested and outside of the chip. So this is a massive improvement that broadens the range of cells that become now accessible to 3D analysis. Another feature of our 3D CoSeedis chip is that we can use the before mentioned distance co-culture. As you all know, for some cells it's necessary to add feeder cells to the cells, and we call that contact co-culture in this case. So the feeder cells are mixed with the test cells, they support the test cells, and together they can form nice aggregates. However, those intermingled cell aggregates will not be separable anymore after aggregation, and we always have a mixed culture. In 3D CoSeedis, however, it is possible to underlay the entire chip with a layer of feeder cells that then secrete growth factor and other protein factors into the media, which diffuses freely through the chip and influences the test cells above in the 3D CoSeedis chip. They do this, have the same effect on cell aggregation as is shown on these panels here. However, it is possible to separate the two cells again after aggregation has taken place. Now, this idea of distance co-culturing, of course, goes a lot further than just simply using feeder cells. First of all, having such a system makes it possible that in a lot of circumstances we can work on the serum-free conditions. So these cells here will only use the feeder cells to support themselves, and of course they can also communicate with each other, and that allows in a lot of cases that serum can be left out of the medium, and in fact we sometimes work only with basal medium. But it goes yet a step further. It also allows us now to build up cell communication systems whereby, for example, cells in 2D can communicate with cells in 3D. We can kind of mimic organ-to-organ communication, and we can even, instead of having the feeders or the second cell type grown in 2D, we can grow those cells in 3D. Latest experiments now have in fact even allowed us to stack two different 3D CoSeedis chips on top of each other. We call that a sandwich approach, whereby now cell aggregates from one chip communicate with the aggregates of the chip above. So that allows us now a new way to approach rather complex disease models and to mimic cell-to-cell or organ-to-organ communication. On the next slide, I simply want to give you an overview on all the different cells that have been tested so far in 3D CoSeedis. This list is continuously growing, but as you can see already, we've tested quite a lot of tumor or cancer-derived cell lines. We also included some blood cell lines here, T lymphocytes and B lymphocytes. Obviously, those cells do not aggregate. However, they sediment into 3D CoSeedis chips and can be grown quite happily in those chips as well. We've also done some mesenchymal stem cells, primary cells, for example, epithelial cells, and so on and so forth. For a more detailed overview, I recommend the following publication, or an accurate and updated list is contained in each of our protocols. Here's an overview on the current product portfolio that we offer. So all our chips, the chip 200, the chip 880, and the chip 680, come as individually packed chips in an aluminum bag. They contain a bit of PVS to prevent them from drying out. They are sterile, and they have a shelf life of 3 years. Furthermore, the chip 880 and the chip 680 we also offer as bulk in those beakers, where there are either 6 or 6 and 12 chips in one beaker. And of course, we offer protocols for various different applications. We have protocols, for example, for histology, volumetric analysis, and we recently published white papers on the use of our chips in HTS or HCS. And that brings me straight to the applications on which I'm going to spend the last part of our webinar today. We have designed 3D CoSeedis as a standardized tool to mass produce spheroids. And the idea has been that the 3D CoSeedis platform is always used as a plug-on to existing downstream applications that then can be used in different areas that are listed here. And maybe I just show you the first application as an example. So the 3D CoSeedis plug-on is used upstream any standard application. And the idea was that we use it as a tool to feed highly homogeneous and uniform spheroids, organoids into this existing standard procedure. In this case, it was immunohistochemistry or immunofluorescence, but always with the goal to not change the protocol or the process of that existing application. Now, for example, here we're talking about immunohistochemistry. So the 3D CoSeedis chip can in fact be used to not only grow the spheroids now, but we can directly embed those spheroids inside or with the chip into paraffin, for example. So it is possible to dehydrate the entire chip, to embed it in paraffin, and then section the chip straight away, which results eventually into an array, microarray type of arrangement of our cross sections through spheroids. Here is an example or a few examples of such an array, how that looks like. They are stained with different antibodies, immunohistochemistry. We also have different sizes of our microtumors, just to show you the variety of what's possible. Important is, however, that this entire process can also be automated, and even the analysis, due to the fact that we have this array-like arrangement, is possible in an automated way. We've also done a collaboration with Cell Signaling Technology, which tested that in-house, both also in cryo. Cryo embedding is as well the same, it's possible as well. So they've done a test on cryo embedding with immunofluorescence, and they've done a test on paraffin embedding with immunohistochemistry. Again, you can see here we have a cross section more or less through the center of a spheroid, whereas here the section is more at the bottom or the top of the spheroid. And here you can clearly see that they were quite satisfied because handling of such spheroids is a lot easier. You don't have to manipulate individual spheroids. It can all be done in the chip, and therefore makes damaging or losing spheroids almost impossible. Another application for which we have designed a plug-on is in the area of clinical research, namely extracellular vesicles and tumors. Here, the 3D CoSeedis platform offers a very nice way to grow and collect conditioned media containing large amounts of extracellular vesicles that later on then can be purified and analyzed in the downstream process. This work has been published about 2 years ago, and in that publication people have shown that indeed it makes a huge difference where the cells producing extracellular vesicles are indeed grown in 2D or in 3D. And the difference is namely that they show a completely different content in microRNA and protein, but also their composition of the entire population of extracellular vesicles in terms of shape and size is remarkably different. More important, though, is the observation that whatever they isolate from 3D is indeed much more similar to what is observed in, for example, blood of a patient. For us, what was the most important part of the work is, however, the recommendation to use 3D CoSeedis as a platform now to collect these highly enriched conditioned media. It is possible to grow up to 880 spheroids under the very same condition, meaning that they also produce very same or similar extracellular vesicles that then can be collected from one individual well, and therefore gives you the chance to have highly comparable conditioned media. Yet another approach or another plug-on comes again from clinical research, but it now concerns tumor growth and relapse. One of those results I've already shown you. In addition, here is just another few scans to show you how this actually looks like. So just to remind you here, we compared treatment of irradiation alone and treatment of irradiation and hypothermia together, and then counted after up to 11 weeks the tumors that popped up again, so that continued growing after a certain period of time in culture, indicating relapse, their relapse, and that's exactly what was compared here. Now, the same thing can be done, of course, not only for hypothermia, but also for a combination of irradiation and chemotherapy. And already by eye, you can see which dosage and chemical combination has an effect by simply look at those transmission scans here. Now, we also spent time on regenerative medicine, and there was some work done on the growth of bone marrow derived mesenchymal stem cells, which we plated in 3D CoSeedis to maintain and expand those cells, which works really nicely. And then the cells have been transferred into media inducing chondrogenic differentiation in this case. And as you can see here, all microspheres did indeed differentiate into chondrocytes, producing very nice cartilages, as indicated in this magnification here, where we see the cells nicely surrounded by cartilage, but also on this side where we stand hyaluronic. And you can see that in the center, it's very nicely formed, whereas the outside is more fibrous still. Now, I would like to go on and spend some more time on the last applications here, where we looked into our compatibility with high throughput screening. Now, in this particular case, it's obviously very important that we design a plug-on that does not change the standard processes that have been developed in high throughput or high content screening. And as an overview, this is basically shown here. So what we have done, we used the 3D CoSeedis Chips 680 on a 24-well platform, delivering us more than 16,000 spheroids per plate. These spheroids can easily be harvested then. For simplicity reason, we've shown that here in a 6-well dish, but it works just the same in 24. So you can see here centralized the harvested suspension of spheroids grown in 3D CoSeedis. That's another magnification of the same thing. And here a light micrograph showing the homogeneity and uniformity of those spheroids. Those suspensions then went into a large particle sorter. We did that in collaboration with Union Biometrica. This large particle sorter could sort individual spheroids into wells of a 384 well plate, which then went into HTS downstream processes. From this stage on, basically the entire downstream process is automated and is fully compatible with what is currently standard state-of-the-art in HTS. We then, and we've published that recently in a white paper together with Union Biometrica, we then looked on how and what do we actually get as a result in those 384 well plates, how viable are our spheroids after the sorting. And here on the left-hand side, you can see the results shown with HT29. I would like to draw your attention to panel D here. This panel shows nicely a red diagonal line, which basically separates viable cells on the top from cells that would have died off during the process at the bottom. And as you can see, all the cells we sorted were perfectly viable, indicating that the entire process gave us 384 well plates that only contained viable spheroids in individual wells, and we repeated that as well with different cell lines from different tumors, such as prostate, for example, or breast, or colon, and there was no difference between all those cells. All cells gave us viable spheroids sorted in 384 well plates. So we now therefore have a process developed that fully supports high throughput screening workflows without changing the current protocol. It provides highly reproducible and reliable 3D constructs in high throughput quantities. We can easily upscale that. I mean, as I said, one plate derived gives us more than 16,000 spheroids. So we quite easily reach HT quantities. And 3D CoSeedis also allows now predictive modeling based on those HT results, because we can deliver 384 well plates that contain viable spheroids in all wells of a high uniformity and a high homogeneity. We've done a similar experiment then to addressing high content screening. Again, this was done with the 3D CoSeedis Chips 680, and it was done in collaboration with biotech, whereby we used their citation 5 system. In this system, you can have, in fact, also you can hook it up to an incubator that allows you to incubate up to 8 of our platforms here, totaling more than 130,000 organoids or spheroids per run. That can also be automated, can be supplied in an automated way with a medium or a compounds. And then we can add those into the citation 5 for automated microscopic analysis, which is possible because the 3D CoSeedis offers a 3-dimensional grid work in which the position of each individual spheroid is fixed. So the microscope knows where to actually take a picture. And again, on this panel, we just show you the homogeneity and the uniformity we achieved in this particular experiment across one chip. The same is also true if you compare different chips or even different platforms to each other. Now, as a summary and take-home message now, 3D CoSeedis is now, I hope you could convince you, an ideal tool to mass produce highly homogeneous and uniform organoids and spheroids. It is highly reproducible. It is unique in its co-culture design, which allows us then to mimic a wide range of disease models in a relatively easy setup that nevertheless can be analyzed in quite an easy way as well. And it is compatible with numerous downstream applications because it feeds into those applications without actually changing their protocols, not least of them, of course, high throughput screening and high content screening. I would also like to quickly spend a minute on where you can find more information on our technology. Now, first of all, of course, we have a website on which you can find tutorials. You will also find the recording of this webinar there, but there are also protocols and other useful documents there about 3D CoSeedis. We have a YouTube channel on which we regularly publish our latest tutorials, for example, and to always be updated and to stay tuned in on our activities. You can connect with us on LinkedIn, where we publish all our most recent news in the area of 3D CoSeedis, but also 3D cell culture in general. And with that impression from our production and development center in Cham, I would thank you for your attention so far. I hope you enjoyed the webinar, and I will now address your questions. The first question I would like to answer is whether it's possible to tune the agarose concentration depending on the cell type to be used. That's a very good question. We've actually tested a range of different cell agarose concentrations in the past, and we have not really observed any differences in the concentration of agarose on cell growth, aggregation, and so on and so forth. In fact, agarose, fortunately, is so biologically inert that it seems really not to matter how concentrated it is. We therefore chosen a concentration that makes handling of chips easy and nevertheless is transparent enough to use it in microscopy. Now, another question is, how do we choose the best format for 3D CoSeedis chips for our application? That's a very good question. As you've seen, we have three major formats, which is the chip 880, the chip 200, and the chip 680. Now, maybe I start from the back. The chip 680 is certainly ideal for everything concerning high throughput or high content. On top of that, it's also ideal for all applications dealing with primary cells or stem cells because it allows cell culture or culturing of lower cell densities per microcavity, and at the same time it also reduces the amount of media that is used per well. The chip 200 or the chip 200 is mainly used for long-term cultures. The individual spheroids are a bit more separated from each other, which means they can be kept in culture for a prolonged period of time without consuming too many nutrients from the medium. And the chip 880 is mainly used, for example, for clonal growth of spheroids or for experiments whereby I need a large number of similarly grown spheroids under the same conditions, such as, for example, extracellular vesicle, but also that relapse experiment that I showed before. Now, one further question was, how does the conical aggregate go into a uniformly round spheroid? Does this happen during harvesting from the chip? Great, this is a question. It's actually an interesting question because cells that do form conical aggregates do not show a tendency to go into or to grow into a spheroid at all, never. What 3D CoSeedis does, however, it acts and exerts enough pressure on those cells. It's sort of like a crevasse in a glacier. So it exerts enough pressure on those cells to push them into aggregation. So even though they do not round up, they still aggregate with each other and form compact cell-to-cell connections, which makes it possible then to harvest those cones from 3D CoSeedis. However, when they are harvested, they still look like cones. So they're not really rounding up. However, we have so far never transferred those cones into, for example, low attachment plates or so, and continued them to grow to see how they would behave later on. All we have done is we plated them in normal dishes, and then of course they show the similar behavior as spheroids as well. They start attaching to those dishes and cells start to move and migrate out of the construct of the aggregate and grow in 2D. Another question is, we are interested to do some transcriptomics and profiling at the end. Is it possible to access spheroids for that purpose? Yes, indeed it is. Harvesting is a very nice way to get access to all spheroids. In fact, we do have HCS customers that use at the end part of each individual chip for different purposes. So part can go into immunohistochemical analysis, a part can go into harvesting and consequently into profiling by doing RNA extractions, for example, and so on and so forth. A part can be used for whole mount staining and so on and so forth. So there are many different ways you can make use of a chip. And the good thing is, because all the spheroids within one chip have faced the same biological condition, they also act as biological replicates, and therefore they give you a very nice statistical readout at the end. Then another question is, can you reuse the chips for multiple experiments? No, the chips have been designed for single use only. Chips are, once they're used, they're in fact filled with cell debris, or sometimes also with dead cells that were transported out from inside the spheroid, and so on and so forth. So it's not possible to clean those chips, and neither is it possible to sterilize the chip again for further usage. So they're really only for single usage. Now, another question is, have you formulated chips from reconstituted collagen or matricial? I think the question here is whether it is possible to coat our chips either in collagen or matricial. Now, this by itself is not possible. The reason is that in order for the cells to grow at the bottom of each individual microRNA, they must not attach to the sides and the walls of the microcavities. The chips have been designed such that no cells can ever attach to the side walls. However, as soon as I start coating those chips, I would lose that advantage and cells would simply grow everywhere. What has been done, though, is that people have used, or they have prepared matrices such as matricial or collagen. They have subsequently shredded those matrices so that they got a kind of a substantiation of tiny little shredded particles of matricial and also of collagen. And then those little particles, they can be diluted, sort of like in a cell suspension, and then centrifuged to the bottom of the microcavities. After that, cells can be added in a similar way as they would be seeded normally onto 3D CoSeedis chips, and they then can grow and incorporate those matricial or collagen chips, and that helps some cells to form 3D aggregates. Some of these extracellular matrices then get eventually digested, others stay there. That depends a lot on the cell type as well, but experiments like that have been done, and they look actually quite interesting. Now the last question for today is, how do you change medium? Do you see the chip as it is transparent? Yes, indeed. medium change is very simple. Our microcavities are quite steep and quite deep, actually. From the chip 200, for example, and the chip 880, the microcavity is almost 2 millimeters deep. That allows you easy medium change without the risk of sucking in the spheroids. So they will stay in the microcavities. And it's also possible to add new medium without dislodging or displacing those microspheres, microtumors. So that is easy. It's also easy to see the chip because the chip acts like a sponge, so it normally gets red from the color of the medium. So it's quite easy to detect the chip, and even if the medium wouldn't contain any indicator, color indicator, then it's still easy to see it because it's not, the concentration of the agarose is such that you can detect the chip quite easily by eye. Later on, when the spheroids actually have grown enough, you can even see the spheroids because they become big enough. So it's even possible on a black background then to actually see the spheroids sitting in the individual microcavities. Now I'm sorry, for time reason, we come now to a finish. If there are any more questions you would like to ask me, please visit our website, go to our team section and send those by email to me. I'm more than happy to answer them by email, or you can simply call us and discuss them on the phone with me. For now, I thank all of you for your attention, and I wish you a nice rest of the afternoon and a good evening. Bye-bye for now.